So today I just wanted to speak to you on some preliminary data that we have on the impacts of exposure to PAHs in bioindicator species, river otters. So this was primarily done through community-based monitoring programs. So instead of showing an outline, river otters are hilarious because you can find all these internet memes on them, but basically what I really wanted to focus on is some of these PAHs. They share many similarities with persistent organic pollutants. Really what I wanted to stress is that they're thought to readily metabolize, and you often don't find them in very high concentrations, and we're finding a different pattern, especially for the alkylated-PAHs. We're finding that these compounds, they readily bioaccumulate, and they're toxic to aquatic organisms, and because they can bind to PM2.5, they have this capacity for long-range transport. So why are we working in the oil sands? Well, I mean, I think there's a number of different studies pointing to an increase in inputs from the oil sands industry, especially alkylated-PAHs. These compounds are a reflection of the end-chain plant terpenes that underwent these diagenic processes to form the bitumen. And basically, a big driver, as you've heard this morning, is TK holders are really driven by two main questions, the communities I work with, and is this food safe to eat, and is the water safe to drink? I wasn't here for 5 minutes yesterday when the question was already pitched at me. So it's a very important question and something that we're really careful in working with communities to answer. Now, I'd like to acknowledge that we don't have the expertise. We're not medical doctors or professionals, but we do collaborate very closely with both provincial and federal health authorities that are well equipped to answering these questions. So why river otters? I'm just showing a pictogram. This is appropriated from the Haida culture. I couldn't find a Dene or a Cree drawing of a river otter, but elders agree that river otters, being a semi-aquatic mammal, they're tied to water. As you see from water flowing through the animal, water is a source of birth and rebirth, a source of energy, very important spiritually. So any animal that's tied to the water for elders and communities is a good indicator of aquatic ecosystem health for these communities. From a Western science perspective, working with mustelids or semi-aquatic mammals makes sense. I mean, they're sensitive to aquatic pollution. They've got relatively small home ranges. They neither migrate nor hibernate, so this means that they're exposed to pollutants year-round. For otters especially, there's been population declines across North America, and these have often been attributed to contaminants, to pollution, especially the pulp and paper, some of the earlier studies. Monitoring river otters is often difficult. The creatures are secretive, they're elusive, they're difficult to capture and recapture. So with the direct techniques of putting transmitters on the animals, it's time consuming, it's really expensive. So we may influence this natural behavior. It's quite invasive. Using shrimp cakes to lure river otters also doesn't work. So what we're doing is moving away from these traditional approaches of measuring contaminant burdens in tissues or using radiotelemetry, and we're using a fecal sampling approach, which is a really useful non-invasive tool to answering some of these questions. So really where I wanted to go with this today is to basically sort of discuss quickly some of these endocrine disruption impacts of these PAHs. PAHs are known as endocrine disruptors. They'll impede various thyroid and hormone regulation pathways through various modes of action, mostly tied to the aryl hydrocarbon receptor pathway. So, you know, there's a concern that maybe with an increase in exposure to some of these compounds and the metabolism of some of these compounds into their often more toxic metabolites, that we could be having an impact on the endocrine system. So these are the sites. Just again, speaking this morning to a large collection, working with many communities and land users through the Alberta Trappers Association especially. All of these polygons are trap lines that we've monitored since 2012, and then with a focus on the surface mineable area right here, so there's two trap lines there. So we've monitored otters, a focus on one trap line in the mineable oil sands, and then one trap line in sort of a more pristine area of the boreal forest. So these two maps here just shows the nature of some of these anthropogenic disturbances. So the top trap line is the trap line that's much more disturbed with various disturbance classes, and the bottom trap line is more pristine, and these dots are all well sites, which are not uncommon on the landscape. So how we do this, we do latrine site surveys. Otters are like cats, they'll defecate in the same spot. The spot is used with high fidelity by members of the population of families. So what we do is we first of all identify the latrine sites, we mark the old feces, we revisit every 24 hours to collect fresh samples. Once we find fresh feces, we swab them, we collect them, and we store them for either contaminants analyses to look at fecal hormone metabolite levels or to get measurements of population demographic measures. Huge amount of work. We do this by working with the communities, by training the communities to collecting samples, collecting tissues for the dissections when they're trapped commercially for the fur trade. And we really work on increasing the capacity for some of these communities to participate in these monitoring programs. And we learn from one another. This is youth in Fort McKay, so sitting around eating pizza and cutting up beavers. So just talking about youth concerns and going through the biology, just fostering this interest in the land and the health of the land that they're so closely tied with. Working with elders, spending time on the land, very important. I've said this morning. So these are just aspects of the monitoring program that we've been really careful in being inclusive and being proactive in our approach. We spoke about including traditional knowledge with some of our Western science. One of the tools that I've been using that I've found really successful is pairing elders with scientists, non-scientists, and even youth. This was a classroom that I did at a local high school with, it was 30 different communities, 2 members of each communities, elders, youth, Aboriginal youth, and non-Aboriginal youth from the high school. So working in teams, answering the questions, the elder providing insight as to what is a normal liver color, how should mussels smell. So by using these indicators, like we heard this morning, by identifying some of these indicators when we're faced with the large numbers of samples to screen, it's really useful for us in targeting some of these different samples and spending more time on them just based on some of these traditional quality indicators. Latrine site surveys, just to give you a few pictures. I like pictures. So latrine sites are really easy to identify, either in the wintertime or fall time. The monitoring is done primarily in the fall and winter during trapping season. So once we find the scat samples, we essentially swab them and collect them, and they're taken back to Ottawa for analysis. Huge number of latrine sites sampled at each trap line. I'm showing 2 here. So this is the lower, the lower disturbance. The lower disturbance trap line right here, so a large number of latrine sites were monitored. This is a trap line in Fort McMurray. So again, many latrine sites, and they're visited every 24 hours. So it's a huge amount of work, and we couldn't do it without the help of our collaborators and stakeholders. I won't go through this. If you have questions, we can talk about it. But basically, the tool that we've developed is a great tool for community-based monitoring programs, and we've recently submitted a manuscript on that, where we've improved the amplification rates of some of these samples. So usually when you're doing fecal swabbing, you're getting a success rate between 12 and 31%. So now we're getting success rates approaching 70%, which makes the tool much more much more viable, a better, stronger tool, and much cheaper now that we're not wasting over 70% of our samples because we can't amplify anything from them. I just wanted to, and I apologize, this is a poor choice of words, impact. Let's call them disturbances. So the mineable oil sands area, an area with more landscape disturbances, more infrastructure, and then the lower impact trap line. So just to show you the number of fecal samples that were collected on each trap line, and then the tissue samples collected from each trapper. So the carcass, when they're done taking their fur, the carcass is usually discarded or used as bait, and this is where we come in and we store the carcass and we use them for contaminant analyses and answering some of the questions we have. There's some QA, QC that goes behind some of these. I basically wanted to draw your attention that we're only able to detect 5 matches between fecal samples that we've been collecting and tissue samples, simply because we're working within the constraint of the fur harvest quota. So each trapper is only allowed to take a certain number of otters, and we don't want to go out and collect otters ourselves. So 5 matches of tissues and feces, so we could look at biological responses in fecal metabolite levels and some of the resultant residue levels in target tissues. There are 7 instances where we were able to match fecal samples for the whole season. So this means that we can get like a time series analysis of responses to a certain stressor, and you can detect that pretty well in fecal samples. Some preliminary results. So just drawing your attention to this line here, it's slightly increasing. This is looking at allelic frequencies, and this speaks to a higher genetic diversity at some of these lower disturbance trap lines on the landscape. So basically the underlying mathematical models to do this kind of work relies basically on a capture, mark, recapture. So similar thing, we're genotyping each individual, seeing how many times it comes back to the area. And with these metrics, we can calculate a whole whack load of different population demographic measures. One of which is, I find interesting, that the high disturbance trap line had a negative, this is the inbreeding coefficient, which means that the otters are inbreeding within their own families at this trap line. What we can do with this data is then calculate effective population sizes at these trap lines. So the number of adults that are successfully giving pups or having pups. So at the high disturbance, at the high disturbance trap line, we've only able to detect 6 of them. Low impact trap line last year was about 450, but with a third year of sampling, it's infinity. It's a very large, very healthy, genetically diverse population is what this means. We've done a relatedness analysis on some of these samples as well, looking at parent offsprings. How many parent and offsprings were we able to detect at these high disturbance trap lines? Only 1, whereas we detected 12 at some of the lower impact trap lines. Full siblings 9 and 5, half siblings 1 and 55. So just again, to give you an idea of the health and the structure of these populations on these different trap lines. I've spoken of PAHs. This is, again, preliminary focus only on the 2 trap lines. This is a sample size of 20. We've collected 101, so this is still a work in progress. No statistical significance between the lower disturbance trap lines and the high disturbance trap lines, but again, seeing a higher proportion of alkylated-PAHs, which is not too surprising, and levels that are higher than other levels that we're seeing in the literature. Usually, if you look at the Exxon Valdez or the BP, or some of the sea otter work that's been done, they're finding levels between 10 and 12 nanograms per gram. Wet weight, we're finding upwards to, when you add the parent and the alkylated-PAHs together, upwards to 800. So again, this is just preliminary, but something that we're definitely looking into. The scat samples were collected, we've taken the scat, we've extracted with ethanol, and we've submitted these extracts to an ELISA assay, and if you have questions, we can talk about this, but basically we can measure fecal hormone metabolite levels. The T3, so I'm sorry for these poor slides, testosterone levels, that's the high disturbance, low disturbance. We're always consistently finding that there's always a higher level of some of these fecal metabolite hormones in the feces. Estradiol and testosterone in this case are a little bit out of context simply because it's not the right time of the season to be collecting these, so they're not really biologically relevant. However, if you look at progesterone levels that are known to peak after fetal implantation or during an estrus cycle, these samples were collected during the breeding season of the otters, and we're finding that at the lower disturbance trap line, we're having much higher levels of progesterone. In these fecal samples, and the high impact, very little. Equal numbers of males and females between different trap lines. This basically sort of validates the genetic analysis that we did, so more healthier breeding at individuals at the lower impact trap line. And this is in support of some other studies that have looked at those changes in fecal hormone metabolite levels through time. The cortisol, we looked at cortisol, the stress hormone, much higher at the high impact trap line than the lower disturbance trap line, and T3 as well. So there seems to be an endocrine effect. We really need to, and this is on this slide, we really need to work on increasing through continued monitoring the number of successful matches between a non-invasive matrix like a fecal sample and a tissue samples to then do correlation analyses and looking at responses, biological responses of some of these pH burdens and some of these endpoints. So on this note, I'd like to thank everybody. It's a huge amount of work and a huge amount of people, so thank you very much.